Okay, looks like we're all set to start. Hello, everybody. A very good afternoon to you all. I am Himani. I'm the brand marketing manager here at Cambrian Bioworks, and I'll be your host for today's webinar. We have an exciting topic for today. We'll be covering what's working in NGS workflows right now and the biggest bottlenecks that molecular labs face as volume increases, and how these challenges can be tackled, especially during the library prep process. If this is your first time joining us, welcome. You'll see a QR code on your screen that will take you to our events page where you can subscribe and stay updated on the upcoming sessions. We started this series to create a practical space for professionals in molecular labs to learn from real workflows, understand what's changing on the ground, and exchange insights that are immediately usable. So far, we've covered topics like extracting high quality DNA for microbiome studies, scaling whole exome sequencing throughput in clinical labs. We've also had guest speakers from organizations like Strand Life Sciences and experts like Dr. Harsh from Institute of Human Genetics. And as always, our goal for today is to leave you with clear and actionable takeaways. Okay, let me quickly walk you through for what we've got lined up for today. We'll start with a quick overview of the current NGS workflows and how they evolve as labs scale. We'll then talk about key bottlenecks that lab face today, especially during the library prep and size selection process. We'll dive into practical strategies to improve consistency, reduce repeats, and save hands-on time during the NGS workflows. We'll then focus on CamSelect NGS, our made in India beads. They help with size selection and how they can fit right into your workflows. Let me introduce you to our speaker of the day. You'll hear from Subhashri. She is the product manager at Cambrian Bioworks. She works closely on product development and application use cases, and she'll walk us through how size selection challenges show up in real NGS workflows and what labs should be paying attention to as they scale. With that, I'll hand it over to Subhashri to get us started. Over to you, Subhashri.
Thank you, Mani, and good evening to people who have joined from India. Good afternoon to registrants from Europe if you've already joined. I'm Subhashri and I take care of Cambrian Bioworks' NGS product portfolio. Thank you for joining us in today's webinar, where we are excited to discuss how streamlining the very process of library construction brings about scalability, reproducibility, and reliability of sequencing outcomes. I know from my colleague Himani about the diversity of our audience today, ranging from students to freshers in the genomics industry, to distributors and people who breathe, eat, drink genomics day in and day out. In the interest of this diversity, I thought I wanted to start this presentation by giving a brief intro about next generation sequencing, also referred to as NGS in short. It refers to massively parallel sequencing technologies that simultaneously interrogate millions of DNA or RNA fragments. Now, by coupling clonal amplification, the traditional method that Illumina uses, which is also called sequencing by synthesis chemistry, with coupled with Sanger's, dideoxy terminator based cycling sequencing chemistry, NGS enables genome scale analysis at resolutions that were not feasible with capillary based Sanger sequencing originally. So this shift transformed sequencing from a gene by gene tool into a comprehensive, massively parallel high throughput discovery platform, which we know as NGS today. To understand why NGS is so powerful, it helps to briefly contrast it with what came before. Sanger sequencing, traditionally used as an approach to sequencing nucleic acids, uh, used to help generate one read at a time, making it highly accurate, but fundamentally low throughput and expensive at scale. NGS overcomes this limitation if it can be called as a limitation by fragmenting nucleic acids and sequencing millions of such templates in parallel, dramatically increasing the throughput while reducing the cost per base. Now, this parallelism is the core innovation that underpins every modern sequencing platform as we know today. So once sequencing became scalable like this, The next challenging thing was to decide what to sequence. Now, NGS is not a single application, but a toolbox for addressing diverse biological questions. These questions could range from identifying single nucleotide variants or SNVs and small indels to quantifying gene expression. I'll use the annotate tool here. Yeah, so quantifying gene expression, such as by sequencing RNA-based templates, to profiling microbial communities and to resolve large structural rearrangements, to understanding cellular molecular functions through such approaches, to understand many variety of things such as host pathogen interaction, disease biology, all towards the common goal, approaching the common goal of improving health and managing disease increasingly better. Now, each question places a different demand or variety of different demands on sequencing approaches, including read length, depth, library preparation, etc. So these demands are closely tied to the type of sequencing technology to be used per se. So broadly, different platforms are there using different read lengths, and they fall into two categories: short read and long read technologies. Short read platforms prioritize accuracy and depth, while long read platforms preserve long range information and prioritize read contiguity. Both coexist today because they solve different biological problems and questions. But choosing between these two approaches is not about technology preference, but rather about experimental intent. So let's look more closely at how these two strategies differ in practice. We start with the strengths of short read sequencing. Briefly, short read sequencing generates hundreds of millions of reads, typically between 50 to 300 base pairs, with very low error rates. Short read sequencing platforms are optimized for accuracy, throughput, and cost efficiency. High depth makes this technology ideal for applications such as targeted sequencing, amplicon panels, RNA-seq, etc. Variant detection, where sensitivity and quantitative precision are critical. However, the short read length limits their ability to resolve, complex genomic regions with highly repetitive structure and also long-range genomic structure. And so this is where long read sequencing, plays its role and was developed to specifically address these limitations. Long read sequencing technologies help you generate reads that can span large genomic regions, capturing full length genes, transcripts, or structural variants in a single read. So this ability makes long read approaches particularly powerful for applications like de novo genome assembly, haplotype phasing, isoform resolution, detection of complex rearrangements, etc. Sequencing depth again determines how confidently variants can be detected or quantified with the long read platform as well, like short read platforms. But high depth sequencing is essential for detecting low frequency variants in oncology and mosaic samples kind of setup. But while moderate depth is sufficient for whole genome surveys or expression profiling, overshooting that depth wastes resources. And insufficient depth again will risk false negatives here, just as in the case of short read technology. But here the real trade-off is lower throughput and higher per base error rates compared to short read platforms. However, the bottom line with either technology is that regardless of read length, um, sequencing performance is ultimately shaped by coverage and depth. So let's look at how sequencing field itself has evolved and come to be to this day, where we are able to choose between different resolutions based on different read lengths. Advances in sequencing chemistry, optics, informatics, etc., have dramatically increased throughput while also driving the cost per base. And these improvements have enabled, recently genomic, real-time genomic surveillance around 2020 during the SARS-CoV-2 pandemic, where millions of viral genomes were sequenced globally to track transmission, emergence of variants, evolutionary dynamics at scale. Uh, so in parallel, long read technologies also lately helped us make possible to complete the first gapless human genome assembly, resolving previously inaccessible regions such as centromeres, telomeres, etc., from the missing from the genome assembled and published back in the Sanger era. Importantly, these advances in speed and cost did not just act in isolation. Increased affordability and improved accessibility of these platforms and wider availability of reagents to use across these platforms um, have also expertise have reinforced each other. Uh, and this convergence has also helped countries like India to participate in large national initiatives such as the Genome India project recently, equipping us with our own genome database to use as reference and to participate meaningfully in large-scale genomics across public health surveillance, population genomics, translational research, etc., rather than remaining dependent on external sequencing hubs. So, but access has increased, but so has the diversity of sample types, entering these sequencing pipelines. So, despite this diversity, the core sequencing workflow remains largely unchanged. Every NGS experiment will follow the same fundamental path, starting from sample collection to extraction of nucleic acids, library preparation, amplification of the libraries, sequencing the libraries, um, bioinformatic analysis of the sequence data, and reporting, the actionable insights. So while sequencing itself and bioinformatics plus reporting, because it leads to actionable data, receive much of the attention. Library preparation is a workflow where most experimental variability is introduced. And there's one reagent that appears repeatedly across library preparation workflows in an NGS lab at critical points. These are the magnetic beads. And magnetic beads are used throughout the NGS pipeline for nucleic acid purification, cleanup, site selection, sometimes even concentrating low mass samples. So magnetic beads are magical reagents that enable rapid buffer exchange, removal of contaminants, enrichment of desired fragment sizes, etc. And as a result of that, directly impact sequencing success. So beads don't just clean, they optimize. So by what we do is in NGS labs, routine NGS labs, by adjusting bead to sample ratios, we precisely are able to control which fragment sizes are retained and which fragment sizes are eliminated or removed. And this control allows us to influence cluster density. These may be jargon for some, but this is suffice it to say that it improves the sequencing outcomes or the readouts from the sequencer. So thus overall increasing the sequencing efficiency. So in practice, beads are a key reagent useful for optimizing library performance across platforms. So, but there are many different types of library with library requirements varying widely depending upon different types of applications that we uh, design or envision for answering different biological questions, such as amplicon sequencing, cfDNA based workflows, FFPE samples, RNA sequencing, metagenomics. All these impose different constraints, different types of constraints on things such as parameters such as fragment size, purity, yield of the nucleic acid, or the available sample for library preparation. For example, let's take FFP and cfDNA as classic pain points. FFP nucleic acids are typically fragmented, chemically modified, cross-linked, etc. Formalin fixation is a routine procedure to process FFP blocks, and which manages to introduce base damage and reduce the amenability of the nucleic acid preserved in the FFP blocks to amplification, which can possibly also introduce PCR artifacts, because PCR is a very important part of a library preparation workflow. And the resulting fragment size distributions of the library are already short and broad, ending up as suboptimally low molarity samples. Okay. So, and low sizes also sometimes. So, these parameters or these properties of nucleic acids extracted from FFP samples pose challenges for things like multiplex normalization before sequencing, before pooling and sequencing. Again, cfDNA is a typical NGS lab pain point. cfDNA is naturally fragmented. It displays a 160 to 180 base pair nucleosomal pattern, highly sensitive to adapter dimers and short fragment carryover, and risks getting easily overcleaned because it's short, and consequently loss of precious sample. So, so the overlap shown here of different sample types and the diversity across different applications highlights a central challenge. Many workflows sharing common cleanup needs, but no two samples behave identically the same. So platform choice, again, there are different platforms, as we already saw, short read and long read, etc. Platform choice again adds another dimension to this complexity. Short read platforms often require tight insert size distributions and aggressive removal of short fragments. Long read platforms, in contrast, prioritize minimal fragmentation and gentle cleanup to preserve, high molecular weight DNA, and maintain the long range integrity. This creates a need for flexible, tunable cleanup strategies, and this is where adaptable bead chemistries become important. And this is when we all need a single tunable bead chemistry that allows the same reagent to be used to support cleanup, left-sided size selection, right-sided size selection, double-sided size selection, whatever you want. And this will also, a single reagent will also help reduce protocol complexity, simplifies the validation exercise, improves reproducibility across workflows, um, sometimes all the way, in our case, all the way up to long read workflows, which you will come to see further slides. This is where I want to talk about our own bead-based, magnetic bead-based reagent known as CamSelect NGS, which is a reagent designed for precision size selection and PCR cleanup across a wide range of NGS library preparation workflows. It enables selective recovery of DNA fragments from 100 base pair to up to 1.5 kb, making it suitable for all kinds of templates, including amplicon libraries to fragmented genomic DNA. In addition to size selection, CamSelect also functions as a robust general DNA cleanup reagent, efficiently removing enzymes, salts, primers, adapter dimers, reaction byproducts, contaminants, inhibitors, etc., that can compromise sequencing performance. And because we manufacture CamSelect beads in our Bangalore factory, we also have control over the manufacturing process, which is why they are ultra uniform in size, um, ranging from ranging between a very tight distribution between 700 to 900 nanometers, um, which allows them to exhibit predictable binding behavior and deliver highly reproducible size cutoffs when bead to sample ratios are adjusted. Importantly, CamSelect is designed to plug directly into existing library preparation workflows and is compatible with common magnetic stands, all types of common magnetic stands, manual processing, automated workflows without requiring much changes in the protocol. This product has been in the market also for about 5 years now and has been widely validated across NGS labs in India and abroad, and the NGS labs, diverse portfolio of workflows. So with that context, I'll introduce CamSelect and how it fits into this landscape of NGS labs. CamSelect NGS is a magnetic bead-based reagent again, designed for nucleic acid cleanup and precise site selection across various types of NGS workflows. By adjusting bead to sample ratios, CamSelect will allow you to support a wide range of applications without changing protocols. Now, this is now there comes some data that we have about CamSelect, how CamSelect beads have been performing across different assays, and how their performances can be comparable or compared to other players in the market. We'll begin here by examining its cleanup performance. Here we compare nanodrop purity ratios before and after CamSelect cleanup. uh, uh, Improvements in A260 and 280, A260 to 80 and A260 to 30 ratios have indicated that there is efficient removal of enzymatic carryover, salts, organic contaminants, etc., which are which introduce key problems in downstream efficiencies. But what's important to notice here is not just the improvement in purity ratios, but the variability, especially if you see here in stool derived DNA samples. Um, Stool samples, as we know, are among the most challenging nucleic acid inputs. They contain complex mixtures. Uh, okay. They contain complex mixtures of polysaccharides, bile salts, microbial debris, sometimes PCR inhibitors. I mean, these are all PCR inhibitors that will, again, dramatically vary from sample to sample. So there are various things that control these variabilities, from how the samples are collected, where they are stored, how they are stored, and how long have they've been in a particular storage temperature, etc. So, that sort of explains this large standard deviation bar, reflecting this inherent unpredictability of stool-derived DNA samples. It is not a technical failure here in this data, but you would also I would also like to draw your attention to this particular improvement in the the wide variability of these set of samples tested here, assayed here. There is no, I mean, there's hardly any standard deviation bar that is visible here. So this are, the reagent that we use to clean up this particular sample, these particular set of samples, has been able to bring down the variability and sort of normalize it to a very common ground. Right. So, so this is something that we have been very happily, boasting about. Again, so it just shows to reflect, high SDs might mean biological and matrix complexity, not just the reagent inconsistency or failure. So what we need is a robust cleanup, regimen across such unpredictable inputs or, biological material. And this is where exactly the bead chemistry must be, made to tolerate variability. And this is exactly how CamSelect beads are positioned strategically, validated across gut microbiome customers that have managed to streamline their sample cleanups, very predictably well. Now, transitioning into the most widely used case, the left side size selection, goes on to show these tape station profiles and electropherograms go on to show that although left-sided size selection removes short fragments such as adapter dimers and primer dimers, while retaining larger inserts for applications such as Illumina workflows. But if you see varying bead concentrations to sample across a wide range of ratios, here you see precision cutoffs here and tightening up of the insert sizes. At 0.4x, which is a very low bead concentration, beyond which Ampere starts failing. Here we see that there is a very, very, there's diminished uh, appearance of the shorter fragments. There's increased abundance of the longer fragments. You would also appreciate these tape profiles or tape traces, whatever you want to call them. It's done on a 100 base pair ladder where on increasing or varying the bead ratios, you would see decreasing the bead ratios, you would see increased selection and increased elimination of shorter fragments, increased selection of longer fragments as you go down the bead concentration ladder. So these are the typical use cases for left-sided size selection, removal of adapter dimer from libraries, and making them compatible and optimal for loading on. One second, mouse is behaving oddly. Okay. Removing adapter dimers from libraries, making them amenable for loading on Illumina flow cells, also other flow cells. Removal of short non-specific amplicons where you have a very tight desired population to use and read out from. Also helps you polish your final libraries just before sequencing to remove other byproducts of PCRs and other reactions. Primer dimer removal, again like adapter dimer removal from cases like amplicon libraries. Uh, Improves the insert size distribution, makes it tighter and cleaner for Illumina libraries. Um, in cases of degraded libraries, increases the efficiency or of their performance because they are by left-sided size selection. You're able to retain the most usable and useful inserts. PCR library cleanup, again, it's one of the use cases that we've already sort of highlighted many times. So these are general use cases for left-sided. This is where CamSelect NGS performs very, very precisely well and accurately eliminates what you want to eliminate, retains what you want to really retain, and at a very, very high resolution. Yeah, so now moving on to two other typical cases of size selection scenarios, right-sided and double-sided. Although the use cases are very few, but these are very, very specialized use cases, and where most beads might, look, unusable or have to be very, very many times validated or customized and back and forth between experiments. But here CamSelect from our data has shown that has enabled right-sided and double-sided size selection very well, and helped us to generate specialized sizes with precision cutoffs to optimize the outputs of the downstream application. Right-sided, for example, helps you to enrich shorter fragments, while double-sided allows a tight size window enrichment. Um, and these workflows are important in very few cases, like I said earlier. Um, Particularly useful for cfDNA workflows where you want to remove where you want to retain these short fragments and at the same time remove traces of genomic DNA from originating from cellular contents. Small RNA sequencing, a special application called degradome sequencing, where the insert sizes are very, very small, very close to the adapter sizes, where you want to remove, extraneous, longer sized fragments and enrich the smaller sized fragments. Um, and of course, if you have a very narrow range of amplicons to select, you remove what is above a certain size, what is below a certain size, such as primer dimers. What is above some maybe some non-specific amplicons are there present above your desired size cutoff. You want to remove that by a double-sided selection. This is a very commonly used case where you want to clean up the adapter and the primer dimers just before loading these or pooling these libraries along with others and loading them on the flow cell. These short inserts or short fragments will alter the clusterability of the main library population, because they are increasingly favored on the flow cell for clustering, and competitively sort of push out the main libraries that are your targets. Now, in this particular case, we have a trace from tape station that shows you two extraneous peaks represented over 7% in total, and which, when cleaned up using the CamSelect beads, have been reduced to a very, very minimal level, which makes it very, very optimal for further uh, downstream use. Now, here we had the opportunity to compare, CamSelect NGS against the most widely used bead reagent, Ampere XP, and which is the industry standard. By using identical input and ratios. Input was 100 base pair ladder in this case, and we saw that CamSelect produced size profiles comparable to Ampere XP. Um, uh, there's a very valuable observation I want to draw your attention to. At widely used standard left-sided ratios, CamSelect NGS performs equivalently to the Ampere XP beads, while at lower ratios, which are not very standardly used, CamSelect reagent or CamSelect beads, as compared to the Ampere XP beads, recover much higher DNA, here, as you decrease the bead concentrations here. This also sort of inspired us to explore further a new bead category, which is a topic of another webinar, which we fondly call as the CamSelect long beads, which allow you to enrich high molecular weight DNA, especially suited for long read applications. So without giving away too much too soon, let me close with how CamSelect supports real world adoption. Our customer went on to compare Illumina beads, Ampere XP, and CamSelect against what we had to offer, and they saw that there was comparable performance and in terms of recovery, percent recovery across these four types of beads. But CamSelect NGS came close to the highest performing CamSelect beads, only negligibly different. So goes on to say that CamSelect is compatible with pretty much everything, manual, automated, all kinds of workflows. We aim to streamline your NGS workflows very, very precisely well by supporting through validation assistance rapid integration into existing pipelines, enabling you to rapidly integrate into your existing pipelines without compromising performance or reproducibility, or having to change protocols, having to change your SOPs, etc. Again, you can reach out to me for any kinds of clarifications, not just restricted to beads, but not limited to beads, but pretty much everything under the NGS sun, including study design, custom size selection, validation assistance for beads and other things, troubleshooting, just anything related to uh, NGS. So I'll end with a quick note on what else we do beyond CamSelect. I joined Cambrian Bioworks just a couple of months ago, which means I still may have the rare privilege of bragging rights before familiarity turns into everyday business. What I've seen here in this time is a portfolio that goes well beyond a single reagent. Um, Solutions spanning NGS solutions spanning across library preparation, automation, workflow optimization, real world sequencing challenges across sample types and platforms. The common thread being across all of these, robustness, flexibility, making sequencing work with the samples researchers actually have, not the 100 base pair ladders, right? So, which are the ideal templates for cleaning up and having sharp peaks at various sizes.
Wonderful. Thank you, everybody, for joining us today. A very big thank you to Subhashri for sharing her insights and experiences. And thank you to everyone who took the time to attend and engage with us. We'll be sharing the recorded session with all of you. We hope the discussion was useful and that you take away something useful that you can apply to your NGS workflows. If you'd like to continue the conversation or learn more about CamSelect NGS, feel free to reach out to us. Thank you and have a great day ahead.